Hello everyone, welcome to online biology tutorials. We will start a fresh topic on Human Genome Project. This topic again is lengthy and important too because there are certain subtitles which we have to study for this topic and question is coming every year on it. First of all, let's know what is Human Genome Project. The name itself is giving you a sense that it will be some project that will be dealing with the human and related to their genome. So genome means the genes present in the human being. So this project was basically meant with counting the number of genes. Genome means the haploid set of genetic material or number of genes present in the gamete, and gene means the normal genes which are present in the somatic cells. So this project was basically meant to count the number of genes and also to do the sequence of the nucleotides. By sequence, we mean that whatever the number of nucleotides are there, what is their sequence? Which nucleotide is present after a particular nucleotide, and this way whole genome will be sequenced. Now there are various subtitles which we have to study in this topic, and these will be why it was known as mega project. What were the goals of Human Genome Project? What were the methodologies that were adopted for this project? And what are the steps to accomplish this project? And salient features, of course, will include the findings of the Human Genome Project. One more thing, in the board examination, it is also important that they will not write the full words Human Genome Project. However, they may give you as HGP. So HGP means they are asking about Human Genome Project. The question may come as give the silent features of HGP. So don't get confused. They are asking about Human Genome Project. And the last topic, of course, will be the application of Human Genome Project. Okay, so we will see one by one. Let's get started with mega project. Why it was known as mega project? So right now we are studying the first subtitle. Okay, now as you know that human being, they have 3.3 into 10 to the power 9 billion base pairs, right? So it would be a cumbersome job if we will do the sequencing of such a big genome, right? That's why it was known as a mega project. Now, one thing more, $3 were required per base to sequence, right? This is also important. How many dollars were required to sequence a base in the Human Genome Project? Answer is $3 or $3. Okay, and that brought its estimated cost to $9 billion. Okay, huge amount of expenditure was required. That is why it was a mega project. Of course, to deal with such a big genome, large amount of scientists were also required, technicians were also required, and of course supporting staff as well, right? That is why this was counted as the mega project. Another important thing is that the people, they were expecting that Human Genome Project would be requiring 3,300 books to compile up the data. In one book, there will be 1,000 pages, and on each page, means on one page, there will be 1,000 characters. Okay, that is why this was counted as a mega project. So this is important. 3,300 books, on each in each book 1000 pages, and on each page 1000 characters. So all these points here, they support that this was a mega project. Another important thing, when the people they began in the starting, they began with this project, they estimated that this project will get over in 13 years. And its foundation was laid in 1990, means it started in 1990. So with 13 years of estimation, it should have got over at 2003. But unfortunately, it ended in 2006, means 3 years later. See, it took the time period of 16 years overall, because chromosome number 1, that required 3 more years to finish it up. Okay, and this got finished in May 2006. Right, so it was estimated that it would be 13-year project, but it ended 3 years later. So 13 plus 3 makes 16. So you must know the starting year, that's 1990, and the finishing year is 2006. Suspected year or expected year of finish was 2003. Now, which were the organization that initiated this project is also important. It is United States Department of Energy, who was the sole initiator, along with National Institute of Health. Okay, National Institute of Health. It is abbreviated as NIH, and US Department of Energy is abbreviated as USD and E. Okay, so these are the two institutes that initiated the process, but later people from other country also donated, because they were knowing that in long run it will definitely help a lot. Okay, so these are all points who initiated, why it was known as a mega project, in which year it got started. Okay, now we are moving ahead on the second subtitle, and this is the goals. Okay, so put a next heading, that is goals. There are important goals for it, like storage, sequence analysis, genes, and ELSI. We will discuss it once by one. Okay, storage means, of course, they estimated that if they will be storing it in the hard copy, it would be requiring 3,300 books, but that would have been again a cumbersome job, and it was not at all easy practically. So they planned that they would be designing some databases which will store the data. Okay, so for storage purpose, an amendment was made later, and databases were introduced which have the capacity to utilize the or store the sequences. Sequencing means, this was the goal, that the entire genome should be sequenced to find out which nucleotide is coming after each nucleotide, so that we can get the whole data with ourselves, which will tell us the sequence of nucleotides in the genetic material. Of course, analysis was their main aim, like which gene was associated with which function. All the genes they had in their mind, they were required to be estimated, right? So they had an idea in their mind that whatever number of genes are present in the human genome, they are going to find it out. And with this came ethical, legal, and social issues. Okay, so here capital E means ethical, capital L means legal, and S means social. So with Human Genome Project, there were certain ELS issues that were associated, and they had a goal that they will be sorting them out. Okay, so if a question comes on what are the goals of Human Genome Project, then you can write they had an aim, they had a goal to have databases for the storage, they had a goal to sequence the entire genome and sequencing and analysis of it, and they had an idea of finding the number of genes in the human body, and with these there were several ethical, legal and social issues that were required to be sorted. Okay, so with this we come on to the methodologies that were adopted for this project. They were two in number. First was expressed sequence tags. This was abbreviated as EST small s. Okay, under this methodology, only the coding sequences they were sequenced, not the non-coding ones. Okay, so in express sequence tags, only the coding sequences were sequenced, whereas in the sequence annotation, which was abbreviated as S capital T, both coding and non-coding sequences were sequenced. Okay, so it was already known that certain part of the hereditary material it is coding and certain part of it is non-coding. In EST, it was like coding part of the genetic material, it was only sequenced, whereas in the sequence annotation, the non-coding and the coding part of the genetic material, it was sequenced. Okay, so with this we have finished why Human Genome Project was known as the mega project, which were the agencies that initiated the project, in which year it started and in which year it ended, the goals of the Human Genome Project, and of course the methodologies. Stay tuned for the next video. In that we will be discussing the method they adopted for this project, the salient features, and of course the applications. Before I leave, I would urge a request to everyone that please subscribe to my channel and share it with maximum people so that each one of you can take benefit. Thank you.